Good afternoon, everyone. I'm Sherry Leong. I'm the president of the Bay Area Taiwanese Biotech Association. Let's switch our focus to the latest trend in biomedical field. It's my honor to introduce our biotech keynote speaker for today, a superstar in the field of biotech and oncology, Dr. Sony Sha. Dr. Sha has over 14 years of research and development experience, spanning biotech and academia in the area of oncology, cell therapies, and immuno-oncology. He has served in leadership roles including chief scientist and CEO, and is a co-founder of Acepodia, a clinical stage biotech company dedicated to developing innovative and high-effective cell therapy for cancer that are accessible to a wide range of patients. Dr. Sha has navigated his journey from identifying preclinical candidates to secure FDA approvals. Notably, he successfully brought his own Berkeley Research cell therapy drug candidate through the process, obtaining IND approval and initiating clinical trials in the US. Today, we are excited to welcome Dr. Sony Sha to the stage. His wealth of knowledge, visionary leadership, and pioneering spirit promise to inspire us in the world of biotech and oncology as he takes us on a journey into further on career treatments through arming T cells with antibodies through click chemistry. Please join me in giving a warm round of applause to Dr. Sony Sha.
Thank you. Hello, everybody. It's my honor to be here. And I think this year I already have more than 10 presentations or speech in the afternoon. They like to have me in the afternoon because whatever delay they have, I will make it up. Okay, so I think we're 5 times delay, so I will make it up. So today I want to first, again, I'm very honored to be here. I'm a Taiwanese kid as well, grew up in Taiwan, and then also go to college in NTU, Taida. I'm wondering who is from NTU? Raise hands. Yeah, I have a good amount of alumni. Yeah, so yeah, Taida is good. And then today, I think I'm more taking off my my mom's room. So I'm a major in chemistry as a chemist from NTU, major in chemistry. But my mom is also graduate from NTU literature. So today, I think I'm probably more of be my mom as a teacher and professor, let you know what we do, be a good literature or professor to describe what you do. I think what I would like to promise you is that after my speech, at least you know little what biotech do or what biotech company is doing, and then why and how we're trying to make the world a different place. So our company is called Acepodia, and then I think those words, if you're not really in our field, if you're not chemistry or biology background, you probably can read all of them. Arming T cells with antibodies through click chemistry, but together they make no sense, right? So you can understand individual of them, but together that means nothing. So today, again, hope I be my mom, and then not a chemist head, but more of a literature, and then showing what we really do in a simple concept. So company-wise, I'll also be honored that my PhD advisor, Carolyn Bertozzi, also the core technology derived from her lab. I was a PhD student in her lab at UC Berkeley. And then we invented the technology now Acepodia used to make anti-cancer drug. Honorably, she and then also the technology has been rewarded Nobel Prize last year in chemistry. So it is definitely honor, but it is also quite good because before the Nobel Prize, every time we talk to the pharmaceutical people, they're asking, what is this? What is click chemistry? What is that? How do you make it? What is your research? What? After the Nobel Prize, no question. Okay. Everything we do, no question. Yeah, but I guess I guess it is the highest honor, right? And then also highest validation of science. So after the Nobel Prize announced, now we get no question from all the Pfizer people, Merck people, and then all these J&J people. Now they don't have question about the science, well, which is good. And then based on that, what is the Nobel Prize really is for last year chemistry? It is a chemistry called click chemistry. So click chemistry, it is really a chemistry that can link two different molecules, molecule A and molecule B, link them together in a very reliable, reproducible, and efficient way. Sounds very easy, right? And then why this can be a Nobel Prize? Because through the years, chemistry actually mostly go into the different directions, making things more complicated. So I can synthesize this molecule. It has 16 carbons, 23 hydrogen. Oh, so I can synthesize another one. Oh, I have 18 carbons, 30 hydrogen, so I win. So it make more and more complicated product become kind of an arm race in the chemistry field. However, the click chemistry is on the other side of spectrum. Instead of making things more complicated, can we use chemistry a simple way, very reliable, although it's just maybe one chemical reaction, but very reliable, very reproducible, can happen in any type of environment, in any type of solvent, and then make things happen. So click chemistry is the chemistry that can do in this. Take analogy, it's like a Velcro. Okay, so Velcro, you have one on this side of clothes, one on this side. If we put the Velcro together, they will bind to each other, right? But the Velcro will not bind to the other parts of your clothes or shoes. So click chemistry has this very special type of uniqueness, and then it becomes Nobel Prize. And then the name is click, but there's another term. Sorry, I do need to bring another term, which is called bioorthogonal. Orthogonal meaning that it will not cross-react. So bioorthogonal is the same idea, meaning that this chemistry happen again like Velcro. Two handles will holding hands. However, they will not affect other parts of your clothes. So they will not affect all the other biochemistry happen inside our body. I think that's the beauty why I joined the chemistry / biology world, because I did get admission from Taida Dianji, but I didn't go. I then won the chemistry Olympia game that happened. So I get to choose any school, any major I want for college. And then I decided to choose chemistry. I actually got admission from electronic engineering. I also can choose medical, be a doctor. But yeah, I think chemistry is I like. And then the beauty is this, right? So the biology, chemistry, they're a complicated system, but they are also very beautiful. And then among this complicated, beautiful system, my advisor, Caroline Bertozzi, invented this chemistry. It's very complicated, very beautiful. However, we have simple chemistry that can link two molecules together, very efficient, very reliable, very reproducible, and then doesn't affect other things that are going on beautifully in the biological system, in our body. So this basically the platform we have. And then, so what we do with it, right? So what we do with it. So so Acepodia and then my research do is that taking this click chemistry, this Velcro system, we link T cells, which is an immune cells that defend cancer cells, defend virus, defend all these pathogens in our body, and then link them with anti-tumor warhead. So that in essence, that's pretty much what we do. So link the immune cells, which is the army of our own body to a warhead that can recognize and then target tumor cells using click chemistry. So this is also basically what we trying to solve. So this, for us, the end goal is to help cancer patients. And then cancer cells, they generate it every day. Okay, so like right now, every time, every day, there are 100 to 1,000 cancer cells generated every day. But we, at least all of us, we didn't have cancer. That's because our immune system, our immune cells, wipe them out every day until they are not strong enough. They either first, oh, sorry, this is not a laser pointer, until at least they are no longer recognize the tumor cells because we grow old, we have a mutation accumulated, there are other factors, so the immune cells cannot recognize cancer cell anymore. And also second, when we grow old, accumulated mutation, there's also potentially T cells, which is the largest army of our immune cells, they get exhausted. So those two factors results in they can no longer control or wipe out cancer cells. Again, cancer cells happen every day. 100 to 1,000 cells will turn malignant every day. They all got checked or wiped out by our immune system. However, when that system become imbalanced, then it become cancer, it become tumor. So what we do is we're trying to address those two challenges, or you can say those two mechanisms happen in our body. So the approach we take are two of them. One is that, as I mentioned, one is the T cell exhaustion, T cell getting old. For that, we have one platform. So for this mechanism, our approach is that we get T cells donated from young and healthy donor, and then use those T cells to supplement for patients' old T cells. In the term, it's called allogenic T cell therapy. Allogenic means we're taking the cells from another individual, treat patients. So this is one platform we have. And then the second is one based on the click chemistry, is that we can link the anti-tumor molecule, which, for example, an antibody, to the T cells using this click chemistry. And then this antibody serves as a warhead. So it's kind of an anti-tumor warhead. Even if T cells themselves can no longer recognize tumor, with this warhead, they can re-engage and then target tumor cells. So this is basically the things we want to do. I think it's biological system is difficult. However, the idea or concept is quite straightforward. In essence, we trying to help patients, provide them a young and robust T cells. In addition to that, those T cells has a warhead, has type of a GPS, they can find, recognize, and then target tumor cells. So this is looking closely, but again, long story short, I think in essence, we were able to use this chemistry called click chemistry, and then get the antibody link to the T cells, and then specifically on the T cell receptors, on the protein, specific protein on the T cells. Again, this is the beauty of this chemistry. With the click chemistry, again, I can the beauty is that like Velcro, right? So when I want to put it together, then I put it together. If it's not a Velcro system, if not the click chemistry, when I generate the whole thing, your clothes is already attached. So you cannot open it or close it. So the beauty of the click chemistry is that I can make this part, I can manufacture this part first, and then this part, and then eventually at the timing, at the stage I like, then I can click them together. And then this type of, this powerful chemical reaction allows us to make what we want, which is put an anti-tumor warhead onto the T cells. And then another part is which T cell we use. Again, I briefly mentioned the T cells we use to treat patient is an allogenic T cells, meaning that it's from a healthy and young donor. So why from a healthy young donor is also quite obvious, because their T cells are young, right? And so be more robust and energetic, not exhausted. So from this young and healthy donor, also another spatial criteria we have for these T cells is they are the one called gamma delta T cells, but that's just a name. It's named after the specific protein expressed on the T cells. But this type of T cells is very unique because when we are newborns, when we are babies, all of our T cells are gamma delta T cells. But when we grow old, like all of us here now, as an adult, 90% of T cells transform to alpha beta T cells. So why we use the gamma delta T cells? Because it's the 100% of our T cell from embryo to newborn, they do not reject mother's cells. So they are a perfect cell type for the allogenic use, because you can get from a donor A or donor B, and then use those cells to treat other patients because they don't reject. That's their design by nature. So these are the two platforms we have. And then this is just briefly how the manufacture flow look like. I do chemistry, right, as you know, so I know this process probably a lot easier than TSMC. But anyhow, so so first we get the blood donated by the donor, and then from the donor blood we can expand this gamma delta T population 10,000-fold. So that's why we can get one donor, we can treat 1,000 patients. So I think that's also a beauty of it, right? So so actually from this one donor, he were able to definitely, with our manufacturer, he will able to help 1,000 cancer patients. So after we can expand these gamma delta T cells 10,000-fold, then we do this click chemistry to get the warhead, those anti-tumor warhead, onto the T cells. We freeze the cells, like the BNT Moderna, you freeze those vaccine, and then you can ship to the hospital. When the cancer patient need a treatment, when their immune system grow weak, and then the tumor become out of control, then they can use the drug to treat those tumors. And then this just a simple comparative landscape. So you may ask, so this is a simple concept, right? You put the warhead, you put the GPS on the T cells. Other people may have thought of it. The short answer is yes. However, here is the difference of what we do and then other companies right now. So so probably from the right to left. So currently, a company A, another biotech company in the US, so what they do is actually quite interesting, and then also very scientific. So what they have done is that if they only use the warhead, okay, no cells, well, we just use the warhead. Maybe warhead can just work by itself. So if they just use the warhead, the response we got, so this is, sorry, another term, the ORR is in our medical field, stands for overall response rate, meaning that the tumor have response to your drug. Also means the percentage of patients have tumor shrink. So only use the warhead, 30% the patient have their tumor shrink. However, when they doing the things we want to do, the same idea, we put the warhead together with the immune cells, when they did it with MD Anderson, the US #1 cancer center, this response rate go up to 97%. Meaning that once you put the warhead, not by itself, but you put it together with the immune cells, the response rate, 97%, meaning 97% of the patient treated with this product have tumor shrink. So this is a significant increase using the warhead or using them together. However, so for Acepodia, we can do, probably the most important is here. So so this one, for what I can do, they want to do the same thing. They want to have the warhead on the cells. However, we have the click chemistry they don't have. So for them, the warhead bind to the T cells just through the we call protein-protein interaction. So the caveat is that this protein-protein interaction is weak. So after they make their product, they cannot freeze and thaw the product. As we mentioned, this is an important cold chain, like BNT, like Moderna, all these vaccines need to be frozen and then shipped out. So for them, the clinical trial they have actually on fresh cells. What's the fresh cells mean? Fresh cells means they are not cryopreserved. So after they generate the cells from donor, they put the warheads on, they need to use it right away. They cannot freeze the cells because when they freeze and thaw, the antibody will fall off. Antibody is the name of the warhead. So so this become problem because to me, this is not commercializable, right? Because it cannot be commercialized because it's almost impossible when the cancer patient go to the clinics, then you find the donor to donate the cells and then doing all this. And then use the fresh cells cannot be cryopreserved. So our strength in this case lies in because we have this click chemistry which form the covalent bond between the warhead, which is antibody or T cell engager, to the T cells so that our final product can be freezed and later thawed. The warhead will still remain on the immune cells, on the T cells. So this is pretty much what we do, and then I think that's also kind of an interesting type of science or technology we do at the interface of chemistry and biology, and then we've been the only one because we possess this click chemistry tool and then also the technology platform. So we were able to generate covalent bond between those warheads and the immune cells. With that, this is our pipeline. So the good thing about our technology is that it can really mix and match. So we can put different warhead on different type of immune cells. Immune cells, the biggest one is T cells, but there are others as well, such as NK cell and macrophage. So our technology platform allows us to mismatch all these different type of immune cell and then also different warhead. Why that's important? Because against different type of tumor cells, then we can have different warhead to target them and then to recognize those different type of tumor. So currently we already have 2 products in the clinical trials and then the third one going into the clinical trial next year. For example, the first one is targeting HER2, so meaning targeting the HER2 molecules expressed on the tumor cells. And then those are for breast cancer, gastric, ovarian, endometrial, those type of cancer. The second one is targeting CD20 molecule on the tumor cells. This is for leukemia and lymphoma. EGFR upcoming is targeting EGFR molecules on the tumor cell. All similar concept, just mix match, switching the warheads. And then EGFR is for head and neck, lung cancer, and colorectal. Yeah, so this is the idea. I think this basically, with the 2 platform we have, we can go from warhead to another warhead and then generate different drug candidates against a variety of different cancer. So with that, this is our team. Again, this never done by a single person, and then I'm very honored and then very lucky to have a very strong team support me on this journey. Thank you. So I think that will be hopefully today I take a good head of my mom, and then we're able to describe to you what we do in more of a simple type of a concept. And then look at, I think I got that 5 minutes back. So I'll hand it to our panelists, which they are all awesome, and then can share you the more deep and then recent development of biotech. Thank you.